It's awesome to be here. I'm excited to talk about this. Is one of my favorite subjects. My background is a little bit different. I am an optometrist, and so I've done low vision rehabilitation clinical work for 20-something years. So very familiar with the patient story, the patient side of things, how assessments occur, at least at a clinical level. And I got into industry about 11 years ago or so and really focused more on RP in the last 8. And so, when we talk about having an assessment to determine whether a treatment works or not, or is effective, we have to understand, the patient's part in all of that. So I have a PhD as well, and that was a little more recent in the last 10 years, and that's actually in higher education. So it's not in vision science. But that gives me a kind of a unique background around trying to understand how to apply a certain model and apply it across many fronts. So today we're kind of in the retina space, or this afternoon, but I'm hoping that what I present today is actually relevant in anterior seg, glaucoma, and lots of other areas. So I'm going to use a couple examples. If we have the time to go through them on a deeper level, great. Otherwise, we'll kind of skim along. The question, does it work? So we hear a lot of presentations, a lot of amazing pitches, and the first thing that I think about is how are you measuring that it's effective or its efficacy? But does it work is a really important question. I'm staying at the Marriott, and I actually, as I was getting ready in the morning, there was a bottle of lotion, and the name of the lotion says, "This works." And I thought that's kind of funny with my talk today. I have very dry skin, and what does it mean that this lotion works? Does it mean it stays on my skin a long time? Does it mean that it relieves my symptoms? Is there some kind of ingredients that have to be included? This actually applies to eye care and ophthalmology as well, right? As we think about, well, who's measuring it, who's determining what that is. What I'm going to talk about is really a little bit more around FDA and what FDA requires. What are the ingredients, or what are the outcomes, and so on. And within that, we have to really think about what is the MOA that you're talking about, this treatment that we're talking about, whether it's a device or whether it's a drug, et cetera. And then what stage of the disease are we talking about? You can't just say that something is for RP or it's a gene therapy for this indication or that. We really have to think about the stage of the disease and why it's actually relevant. And then how does it impact patients? Because if you just say, oh, you know what, microperimetry, that's the endpoint of choice. Well, that may be a popular word that's going around, but is that for AMD? Is it for early AMD? Is it for late stage AMD? It really matters. It's really hard to do microperimetry on a particular group of patients. I'm not going to talk about microperimetry today, but it does tend to be a hot one. So here's my model, all right? So first think about your therapy mechanism of action, as I mentioned. So my role, I'm actually head of clinical for Samsara Vision, which is an implantable telescope. And that's for late-stage AMD, and it's actually an anterior seg, an IOL type of a device that goes in cataract surgery. And I'm not here to really talk about that, but this is an example where you say, well, okay, what's the indication? Late-stage AMD. These patients are legally blind, both eyes, et cetera. Disease stage, as I mentioned, late stage. So they have a central scotoma. So now let's think about their visual function and also what I'm going to call a functional vision. So some of these terms you may be used to, some others you may not. The visual function is it visual acuity affected, contrast sensitivity, visual fields? Those are visual functions. We can measure those. Functional vision is how does it impact the patient, their activity of daily life, their interaction in society, and so on. So in the case of late-stage AMD, visual acuity, contrast sensitivity is impacted, visual fields actually, central visual fields, but also their reading, their ability to see faces, their ability to just take care of themselves, remain independent in the home. So a little bit further into this then, visual acuity. We think of that as, the main endpoint. FDA's told us it's 3 lines of acuity. This is what we need. But that gets difficult when you're trying to talk about, just helping a disease stay stable. In this particular case with this device that I was just mentioning, well, you get at least 3 lines of acuity, check. But how do we know it actually impacts patients? Well, we want to know that it's actually changing their life, that they can see their grandchildren again, and so on. And so it may be that it's important, it's more than just acuity. But let's get it over the line, let's get it approved. That's what's critical, right, as we're talking about development and so on. So let's go on to another example now. So that was BCVA. We're super used to that. 3 lines. Maybe we can get away with less if we have kind of totality of the data telling the story. So let's talk about retinitis pigmentosa. This is a figure that a friend of mine had created, and I've kind of used it over time to kind of show you might have some tissue damage that you're trying to show some change, whether it's the atrophy, et cetera. But in RP, let's think about the visual function, as I mentioned, that's impacted. Constricted visual field, decreased acuity later in the disease, contrast sensitivity, glare sensitivity, color vision maybe. Those are visual functions, as I mentioned, and then you have functional vision, which is, night vision, dim lighting issues, going indoor and outdoor from different lighting. Reading can be affected later in the disease as acuity is affected. Glare, depression even, that's at the person level. And then, of course, at the society level, we get even bigger issues in quality of life and so on. But let's just say in our clinical trials, we just choose visual function, maybe a little bit of functional vision. So let's take another example within RP and dig a little deeper. Let's say your MOA is gene therapy, and it's some kind of repairing protein synthesis, and it's related to photoreceptor function. That's really important because we need to understand photoreceptors. Are we talking about rods? Are we talking about cones? Are we talking about both? So now you get it a little bit more. Is this particular example, I'm just making something up, but early to mid-stage, let's say it's affecting early to mid-stage. Well, that means that acuity is not impacted. So what are we going to use? Well, visual field's impacted pretty early. Night blindness is impacted pretty early, so maybe we can focus on those. And then maybe it's mobility during the day or night, depending on how large that visual field is. So that's our visual function, our functional vision. So I'll go a little deeper here. So what are those visual functions, as I mentioned? Well, contrast sensitivity is actually impacted pretty early on. Visual field is in some way or other, whether it's actually impacting their function or not, and this nyctalopia or night blindness. So let's just use those as examples. There might be other things impacted as well, even color is impacted a little bit early. And what do we know about tests that are already out there and developed? So never start from scratch. Let's find out what everybody is using out there, what are the publications, and then let's review existing FDA advice. What do we know? Well, contrast sensitivity, if it's a secondary endpoint, there's been some guidance, maybe 0.45 log. I like to talk about log because you get different units and it's hard to compare across tests. But log is a good one. And generally, if we're talking about log units, 0.3 log is a doubling. So in visual acuity, we expect 3 lines. That's a doubling of acuity. So going from 2400 to 2200 is meaningful. This is what FDA has told us. So we want to review that existing device. For these mobility tests that are out there, usually 2 light levels is the guidance. But how much is your light level separation? That's important to know. So review what's already out there. But do you think you're done? Let's dig a little deeper. Don't just take your easy endpoints or things people are talking about. When I first began this work, it was like, you've got to do a mobility test. All right, well, mobility tests are out there. Well, no one was letting me use their mobility tests. It was really early on, the days of Luxturna, and they were working on their own thing. Can we borrow it? Nope. So what do? We have to develop one? Well, maybe we do. Let's dig deeper. Is that the right thing for your indication and for your stage of disease? And then here's a really important one. Do you have the right people on your team? Because sometimes you go out and you talk to an expert in the area, and maybe that expert is just so used to lab work that they can't really apply it to a clinical trial setting. It's not realistic, it's not practical, and so on. So we'll talk a little bit more about that in just a second. So who are those endpoint experts? So what I did when I first started this work is I'm not an expert in these things. I know what impacts patients, but still, in clinical trial work, I was no expert. I had done some publications, but it was academic stuff at schools of optometry and so on. So I found every expert I could find that I kind of heard of or knew of and who had published, looked at literature, said who is an expert in contrast sensitivity, who is an expert in visual field, even if it's glaucoma visual fields, who's an expert in the mobility testing, whatever it was. So put together a little panel, and this is where it's really important that your leaders help you with this. Who are the collaborators on your team? And then, as I said, they might be used to kind of a lab setting. So how do you now apply that to kind of a practical approach to these patients? If you're working with a CRO and it's early on, make sure the CRO either knows your area or, better yet, maybe they just stay out of your way. You provide the expertise, and you get involved as a sponsor. They don't love that, but it's necessary sometimes in these rare diseases and these tricky populations. Now, you have the endpoint. Do you need to modify that endpoint? Let's start there. Why start from scratch and validate a device, an endpoint? Let's just modify it. So I call this qualifying. I've borrowed this from somebody else in regulatory. I thought that works. If I need to validate it, let's still not start from scratch. Let's still start from what other people have done. It's way easier to make an argument for an assessment when you have a track record in the publications, even if they're just academic. And then collaboration. FDA really wants to work with you early on. I've done a lot of interactions with the FDA, and they love it. They want to help you. They'll often say, sounds good, give me more. Sounds good, Bring some more to me. I'm like, well, aren't you going to direct me as to what tests I need to do? Go do the work and then come back. Okay, let's figure it out. Rigor, rigor, rigor. This is a very important word. Don't just take a test and say, you know what, I think patients are going to do great on this. Let's just put it in our study. Let's see how they do. And then let's go to the FDA and say, look, our patients are improving on it. Well, did you have a control group? Did you have some rigorous exploratory work where you did some test-retest reliability and you actually know how much change on that test is meaningful beyond just patients having a bad vision day? That happens in low vision, bad vision days. That will throw you off. So rigor has to occur. I actually like having my own endpoint facility where I set up this place, I have a team, and they're doing the testing with my study population. Test it out. So even if you know this has worked for this other program, I want to feel it myself. Are the patients that we're identifying and targeting for our pivotal study, how are they going to do on these tests? Do we need to add an extra visit in there in case they need some practice? What is it we need to do? So let's go a little more into one example, contrast sensitivity. I'll use a couple examples if we have the time. Contrast sensitivity, well, you have Pili Robson that's out there. So, we review the literature. What is contrast? Well, we know, low contrast impacts mobility, fall risk, all sorts of things. Reading. So it does cut across all things that are meaningful, a lot of things that are meaningful in patients' life. Well, tests that are out there, Pili Robson, and you got the QCSF. So this has gone through a couple name changes, but this monitor-based test that presents actually a curve for you. What does the FDA say about those? Well, you see a lot about Pili Robson. It's quoted quite a bit. What do they say for change? This is a term now I'm going to start to introduce, and I'm sure you've heard it over and over, but it's so important. Clinically meaningful change. They will always come back. Even if you say, this is the test I'm going to use, and this is why I'm going to do it, you have to propose, what is the clinically meaningful change in my population, and how am I going to measure it? And if you have sound science and reason with the FDA when you have those conversations, they usually will say, that sounds good, or let's do a little more in this area, et cetera. So in the Pili Robson, I've heard about 0.45 log, 0.15 is each line, so about 3 lines. It's interesting, 0.45, a little more than our doubling that was 0.3 in best corrected visual acuity. Maybe there's a little more variability, and that's why they're kind of setting the stage a little bit higher. But I don't know if that's really set in stone, so you go with what you think and justify it. So do you already know the answer to your change, or do you need to establish it? So how much change is clinically meaningful? How am I going to measure it? And so for me, this really comes down to variability and noise in these patients. So when they come in, if they come in on day 1 and they come back a week later or a month later, not so long that the disease has progressed, but within a certain time period, that's usually maybe up to 3 months depending on the disease, what is their test on day 2 versus day 1? Are they just improving on the second time that you've tested because now they're used to the test and psychologically they're more psyched? Or are they actually having some visual fluctuations? You have to take those things into account and make sure you're working through them. Is there a practice effect? Are you spending enough time on the first day so they're really getting comfortable? I think patient psychology is critical, especially if we're talking about inherited retinal diseases or rare diseases or just anybody who's visually impaired. They are psychologically more fragile than maybe your 20/20 patients. And so being able to put them in a comfortable environment, reassure them, again, this is why I kind of like a separate testing facility. Now, if you say, well, how do you do a testing facility in a large pivotal across the country? Well, maybe you have 2 or 3, and you base them in different geographies and so on. It's not crazy, and it might be worth your time and expense to do that, just so you have a little bit more control and understand your patient population and really can predict better what's going to happen. Do you have, for your test-retest variability, do you also have it linked up with a PRO? Is there something that patients are saying, these are difficult for me, and those responses are correlated maybe to their responses on the test? That's very helpful as well. What is clinically? Because you can just have change. So if I say, well, the patient varies 2 units from test-retest. Okay, and then you look at confidence intervals and you do some stats around that and you say, it's likely that if we have a change of 3 units, that's really meaningful. Well, it's statistically meaningful maybe, but is it clinically? So FDA is still going to ask you, but is that clinically meaningful? How do you know? Trust me. No, it doesn't work. You got to have a little work there. So that's where PROs and some other things can come in to help you say the trust me part, understanding that it really does matter to patients. Patients might say, I just want to stay the same. I don't even care if you get improvement. That's fine, but if you're going for an actual intervention, a shorter term study than just tracking things over time, then you're gonna have to improve vision, and how do you show that it's beyond variability? So let's go in a little deeper with a contrast sensitivity example. So we review existing advice, If you need to develop something, so let's say we need to develop it for a primary. We talked about contrast sensitivity maybe as a single spatial frequency size, that's Billy Robson. But when I talked with FDA about using contrast sensitivity, they told me you really need 3 or more sizes of targets. Pele Robson just has 1 unless you change your test distance. So how do we do that? So a good partner of mine over the years has been Adaptive Sensory Technology. They're the ones who created QCSF. They're here at this meeting today, I think, and good friends. And really we say, okay, they have a test that actually has more than 3 spatial frequencies in their QCSF, but it's a letter acuity, not acuity, but it's a letter test. I found for RP that was really challenging for patients to identify a letter if they have very small visual fields in the advanced stages. Even if they're kind of earlier stage with visual acuity, if their field's pretty small, it's very hard for them to scan and see a letter, and so they have to do letter identification plus contrast. It's a challenging one. So what if you develop, instead of letters, you develop gradings, something like that? So that means you've got to actually do some studies to say, okay, gradings have been done. People have done gradings back in the 60s, 70s, 80s. There's a world of vision scientists out there who published, again, 10 patients, 12, whatever in their labs. But that's some good groundwork to start from. What has been done in gradings, and then how can we apply our modern testing algorithms? QCSF also uses a Bayesian calculation. Pretty cool, because we're used to, in acuity and so on, kind of these stepwise things. This is a time saver because it does a certain process of kind of smart predicting. And so we can do less trials, and it takes patients maybe 10 minutes to do a test versus a true step up and down process that might take about 15 or 20. And that matters for patient fatigue when you've got hours of testing for these patients on a battery of things when we're trying to explore stuff. So development, that can be a way to do it. But you've got to do the studies. Even if you're just doing qualification, you still should do something in your population, maybe a test retest, so you have an idea, again, about this clinically meaningful number above the noise. So here's an example of just a patient with, on 2 different visits, and actually 1 visit and 2 tests. So that's you can do it on the same day. You want to know if you did a couple trials, would they come out the same on the same day? And then how would they be, another visit, let's say a couple weeks later. So validation activities, if you are going to validate, a little more work, but it doesn't have to take you 5 years. You could do this in a couple-year period of time if you're smart about how you set it up and use work that's already been done. So what's been said, there's analytical validity, clinical or content validity. There's some components that go into this. When I first started this, I was like, how do I validate? What do I do? So again, team members helped me, started to work through this, but this clinically meaningful thing is really important. Spend a couple minutes here at the end on mobility testing. This is, again, as I mentioned, this has been out there. You have the Luxturna one, very famous and so on. When I looked at that test, I was happy that they did some pioneering work. It was very exciting to have something that was functional and taking patients' movement and abilities into account. Now, how much do arrows translate to actually navigating across the street and so on? Well, that's debatable, but at least they're not just sitting doing a in-instrument test or something like that. And when I looked at their test, again, we couldn't borrow it at the time, they were kind of working on their own thing, and what I had from the publications was they weren't even light levels, and there were some things about it that needed some tweaking. So I helped JSITE develop a mobility test, and we made really even-spaced 0.3 log unit step levels. We looked at how much change and variability. We did a study with, usually for validation studies, you don't have to have a huge amount. Anywhere between maybe 15 and 20 patients in a particular cohort at least demonstrates the concepts and is able to kind of at least give you some background to go in with the FDA and so on. So this is just an ARVO publication a few years ago where we did some of that work, and we didn't publish a paper on it or anything, but was used in their phase 2 trial. Now, sometimes you develop all this and you find that as you're learning about your treatment, that maybe it's not hitting exactly the MOA. So in Luxturna, they were really, rods were really important to them, and so that test worked well for them. But maybe JSITE or other groups or whatever are using a different MOA, so maybe that's not applicable to maybe their final pivotal study or so on. So you circle back and you look, okay, now I've learned a lot, maybe it's relevant, maybe it isn't. I'm working now a little bit. Now, Samsara Vision is my main, I'm head of clinical there and learning a ton and doing some neat work there. But I still keep my hands a little bit in this IRD space, and there's an area of wayfinding. Wayfinding is really an orientation and mobility term, which is like, how do I get from here to there? Maybe I use some audible things, maybe I use some different clues, and vision's part of it. So wayfinding is not exactly the right term, but I kind of like it because if you're pretty advanced in, so let's say you have these optogenetics, ray therapeutics, for example, and you've got something for a profound visually impaired patient population. They're off chart, they're beyond 2800, they're really in this realm of maybe count fingers, hand motion, and beyond even. What do you do for those? Well, maybe we do some of these grading visual acuity and contrast sensitivity, but maybe we use something like wayfinding where they just have to see a little path going across from point A to point B, and we very simply grade them on a pass-fail kind of a level. So this is an area that's been starting to develop. I think Street Lab is doing some of this work. So there's lots of neat groups that are pioneering and working and developing. What about virtual reality? I would say if you do VR, start with physical first, establish everything you know in physical, and then move to VR after that, and then you have something to compare it to. But be working with the FDA as you go on. So in summary, know your therapy, what's the MOA, even if it's a bit of a black box, where do you think that it's really targeting? And what disease state are you looking at? What stage of the disease? What visual function is impacted? What area of vision might be improved that really matters to patients? Identify those tests, tweak them, revise them, use a separate facility, have a good team that goes together. Use your KOLs, but make sure you're focused in some different areas in those KOLs as well. Don't throw out low vision optometrists. They have an interesting background in knowing patients, so they might be a help on your team. Pick the right team lead, pick the right CRO, be very hands-on. Maybe you need to modify that endpoint, maybe you need to create a new one, but don't start from scratch. Collaborate with the FDA early, ongoing, and use rigor in qualifying or validating those endpoints. That's it, thanks.